If you look at the genome of a 7-year-old with leukemia, logic sort of suggests that you should be looking at a relatively quiet landscape, right?
Right. I mean, you'd think so.
Yeah, because they haven't endured decades of exposure to, like, UV radiation or industrial chemicals, or even just the constant cellular wear and tear that degrades adult DNA over time.
Exactly.
Yet when you actually zoom in on those pediatric cells, what you find isn't some pristine, untouched genetic code. It is an absolute war zone.
There really is.
Entire chromosomes are just shattered, fundamental tumor suppressor genes are deleted, and the weapon causing the majority of this destruction is, paradoxically, the child's own immune system. So welcome to the deep dive. Today, we are fundamentally changing how you view childhood illness.
It's a huge paradigm shift. We have operated for a really long time under this assumption that pediatric oncology is just, well, adult oncology scaled down.
Like it's just a mini version of the same disease.
Right. We assumed the mechanisms of mutagenesis were inherently similar, just maybe accelerated or sparked by different initial conditions. But the data from the 2026 Greenhalgh et al. Study in cancer cell, it completely dismantles that view.
And they did this by looking at a massive data set.
Massive. They compared 1,616 pediatric cancer genomes across 16 major cancer types against 2,203 adult cancer genomes. They basically mapped the entire structural variation landscape and proved that the pediatric cancer genome is a fundamentally distinct biological environment.
Okay, let's unpack this for you listening. Their entire focus here is on structural variants, or SVs. Because we spend so much time in genomics hunting for single nucleotide variants like those little localized point mutations, the typos.
The single letter changes, yeah.
But structural variants are a completely different scale of chaos. If point mutations are typos, structural variants are when entire chunks of pages are ripped out of the book, duplicated, flipped upside down, or just glued into totally wrong chapters.
That's a great analogy. Deletions, duplications, inversions, translocations.
And in pediatric cancer, these SVs account for over 60% of the driver variants. They aren't just background noise, they are the primary engine driving the disease.
They absolutely are.
Right.
And to go back to your opening thought, that initial hypothesis that a child's genome should have a lower structural variant burden, it actually does hold up, but only if you isolate your view to brain and solid tumors.
Oh, interesting.
Yeah, in those specific tissue types, the pediatric variants exhibit a 6 to 16-fold lower SV burden than their adult counterparts. So that aligns with the idea of a quote-unquote quiet baseline.
But the moment you pivot to hematological malignancies, the blood cancers, that rule just completely vanishes.
It disappears. The pediatric structural variant burden in blood cancers is virtually indistinguishable from adult levels. We are looking at this extreme tissue-specific variability.
I mean, how extreme is extreme?
The variance across those 16 pediatric cancer types is roughly 100-fold.
Wow, 100-fold.
Yeah. At the absolute peak of this distribution, osteosarcoma sits with a median of 249 structural variants per genome.
249 massive structural changes.
Right. While on the opposing end, things like acute myeloid leukemia, AML, or Wilms tumor, and low-grade gliomas, they harbor a median of 5 or fewer.
So a 100-fold difference raises a really critical mechanistic question for us. What allows an osteosarcoma cell to tolerate and accumulate 249 massive structural aberrations, while a Wilms tumor is driven by just like a handful?
The answer there lies in the temporal accumulation of damage. It's heavily tied to the age of diagnosis and the underlying germline genetics.
Meaning what they inherit from their parents.
Exactly. In 6 of the studied pediatric cancers, including adrenocortical cancer and retinoblastoma, the structural variant burden actively scales with the patient's age at diagnosis. And to ground that in context for you, we only see that age-correlated scaling in 2 of the 27 adult cancer types they analyzed.
Wait, let me make sure I'm getting this. Because adults are accumulating damage from external environmental factors over decades, right? Whereas these kids are accumulating damage from an internal systemic failure.
Precisely. These age-correlated pediatric cancers are heavily driven by germline mutations in canonical cancer predisposition genes. Think of genes like TP53 and RB1.
The famous tumor suppressors.
Right. When a child inherits a compromised tumor suppressor like that, the foundational failsafe is removed basically at birth. Every subsequent cell division is a roll of the dice without a safety net.
So the damage just snowballs.
It allows structural damage to compound exponentially over the first few years of life.
Okay, let's take a quick second to anchor this for you, the listener, before we dive even deeper. We've established 2 critical baselines here. First, pediatric solid tumors have significantly lower SV burdens than adult tumors, but pediatric blood cancers match adult levels.
Right.
And second, in specific solid tumors, this burden scales with age due to those inherited germline mutations. So the question I want you to hold in your mind as we move forward is this. If blood cancers in kids exhibit this massive structural chaos, but they don't rely on those inherited TP53 or RB1 mutations, what invisible force is actively shattering their DNA?
To find that invisible force, we have to examine the mechanisms of cellular destruction. And we should probably start with the most violent one they found, which is chromothripsis.
Chromothripsis. Just the word sounds aggressive.
It is. It's a catastrophic, localized event. Basically, a single chromosome, or sometimes a localized cluster of chromosomes, essentially shatters into dozens or hundreds of fragments all at once.
Just boom, into pieces.
Right. And then the cell's repair machinery panics and scrambles to stitch them back together via a process called non-homologous end joining.
So it's the equivalent of taking a highly classified 500-page operational manual, running it through a paper shredder, and then desperately taping the strips back together in the dark.
That's exactly what it is.
And the primary danger isn't just that you're missing text, right? It's the chaotic new sentences you form when paragraph 4 from page 12 is suddenly taped directly to paragraph 9 from page 400. You instantly create rogue regulatory elements and novel fusion genes.
Exactly. And that mechanism drives up to a 21.8-fold localized increase in the structural variant burden in those affected samples.
That's a massive jump.
Huge. And the researchers identified this chromothripsis in up to 14.9% of pediatric solid tumors. So it's not a slow accumulation. It is an instantaneous, catastrophic leap in tumor evolution.
And that brings us back to TP53. Because if chromothripsis is the paper shredder, TP53 is basically the system administrator who usually monitors the network and shuts down the server if core files get corrupted.
The guardian of the genome.
Right. In pediatric brain and solid tumors, losing TP53 correlates with the most massive spikes in these structural variants. Osteosarcoma, which we said was our peak SV cancer, operates with a 91.1% TP53 mutation rate.
And adrenocortical cancer is right up there at 80%.
The correlation is undeniable there.
It is undeniable in solid tumors. But, biology always forces us to account for outliers. Take rhabdomyosarcoma. It presents with a remarkably high median structural variant, burden 52 SVs per genome. Yet it harbors TP53 mutations in only 11.1% of cases.
Wait, so it achieves this massive structural chaos while the system administrator is ostensibly still sitting at their desk?
Yes, which proves there's always an exception. But that exception brings us directly to the blood cancer paradox you mentioned earlier.
Right, because if TP53 mutations don't explain the high structural burden in pediatric leukemias, we are looking at a different mechanism entirely. We are looking for an active, aggressive agent that is tearing the genome apart.
And the researchers actually found the signature of that agent. They did this by mapping the intertumor SV distance distribution.
Meaning they just measured the physical distance along the DNA between where the breaks were happening?
Exactly. They measured the physical genomic distance between the structural breaks. Now, in adult cancers, that distribution forms a single predictable peak. The SV breaks occur around 2,000 base pairs apart on average.
But the pediatric landscape breaks that mold completely, doesn't it?
Oh, it splits into a bimodal distribution.
Two peaks.
Two peaks. You see one peak at 12,000 base pairs, and then a second massive, entirely unique peak at under 100 base pairs.
Okay, a sub-100 base pair distance. That is not random chromosomal shattering. That's way too close together.
No, it's not random at all.
That is a signature of highly localized, hyper-precise molecular targeting. It indicates that something inside the cell is making these surgical, concentrated cuts in very specific genetic neighborhoods.
What's fascinating here is that sub-100 base pair peak led them to identify 332 recurrent structural variant hotspots. 332
specific locations.
Yes. These are overwhelmingly microdeletions, and their prevalence is just staggering. They are the driving force in 91.5% of B-cell acute lymphoblastic leukemia BAL, and they're similarly dominant in TAL.
So the mystery deepens for us here. What protein or what complex has the capability and honestly the mandate to make highly precise localized cuts in the DNA of a developing white blood cell?
Well, the prime suspects are the RAG1 and RG2 enzymes.
Okay, break that down for us.
So in a healthy immune system, these recombination activating genes, RAG for short, are essential. They mediate a process called VDJ recombination. Their native normal function is to purposefully cut and splice gene segments.
Wait, they purposefully cut the DNA?
Yes, to generate the vast repertoire of antibodies and T-cell receptors, we need to identify new pathogens.
Oh, so they are the immune system's diversification engine.
Exactly. They locate specific signposts in the genome. These are called recombination signal sequences, or RSS. Specifically, they look for a canonical 7-nucleotide sequence, 5' CACAG-TG3'.
So they see that specific letter sequence, CACAGTG, and they induce a double-strand break right there to shuffle the genetic deck.
That's how it's supposed to work.
Yeah.
But in pediatric allo, the data reveals these RAG enzymes are going entirely rogue.
Engaging in catastrophic friendly fire.
Exactly. They begin binding to RSS-like sequences that are located not in immune gene regions, but adjacent to crucial cancer driver genes like RB1 and CDKN2A.
So they actively snip out the tumor suppressors.
Yes. They even target and delete their own RG1 and RG2 loci. The study found that 88.7% of BAL and 80.4% of TAL samples harbored at least one of these RG-mediated hotspot structural variants.
Okay, here's where it gets really interesting. If RA is cutting outside its designated zones, my immediate assumption as a listener would be that the genome mutated, right? Like you'd assume the DNA sequence near those cancer genes accidentally mutated into that perfect CAGTG signpost, basically tricking the enzyme.
That would be the logical assumption, yes.
But the data entirely refutes that.
It does. The researchers evaluated the targeted sequences using something called recombination information content, or RAC scores. These scores basically quantify how closely a sequence matches the ideal RSS target.
And the scores were low.
Exceptionally low. The non-immune regions being actively cut by RAG in these leukemias barely resembled the biological signposts R8 is actually designed to recognize.
Think of it like an over-aggressive file compression algorithm on your computer. It's designed to search your hard drive for temporary files ending in .tmp and delete them to save space.
Right.
But suddenly, it just starts ignoring the file extension entirely. It starts deleting core operating system files, completely ignoring the fact that they don't match the search parameters it was programmed with. Why is the algorithm ignoring its own code?
Because the targeting isn't driven solely by the primary DNA sequence.
Ah.
Those low RIC scores suggest that 3D chromatin accessibility is heavily influencing the RAG enzymes.
What does that mean in practice?
Well, during lymphocyte development, specific transcription factors physically open up regions of chromatin to regulate cellular differentiation. The DNA physically unwinds.
Like unspooling a tightly wound ball of yarn.
Exactly. And if a region harboring a critical tumor suppressor is physically unwound and exposed within the 3D architecture of the nucleus, a hyperactive RA complex might initiate a break just based on spatial proximity and physical accessibility.
It effectively ignores the poor sequence match just because the DNA is sitting right there, wide open.
It's opportunistic.
Okay, let's pause again and synthesize this second major revelation, because we are about halfway through, and this is dense stuff.
It's a lot to take in.
So we've seen that high structural variant burden doesn't always require a catastrophic failure of TP53. In pediatric leukemias, the damage is driven by a bimodal distribution of SVs, peaking at under 100 base pairs.
Right.
And this hyperlocalized damage is the work of rogue RAG enzymes, the body's own immune diversification tools. They are exploiting the 3D openness of chromatin to delete tumor suppressor genes, regardless of whether the sequence actually matches their target.
Which is just a brilliant summary.
So if you're listening to this, ask yourself, if the immune system is fundamentally hardwired to break DNA in order to function, is pediatric leukemia an anomaly? Or is it an inevitable statistical byproduct of our own survival mechanisms?
It's a haunting thought, because it frames cancer not as a foreign invader, but as an inherent systemic vulnerability. And that vulnerability isn't static. The structural variants we are discussing, they don't just initiate the tumor. They serve as the continuous engine for its ongoing evolution.
So the tumor keeps changing.
Constantly. The researchers actually transitioned from looking at static genomic snapshots to spatiotemporal tracking. They sequenced 13 pediatric patients longitudinally, meaning over time, from diagnosis through to relapse or metastasis.
This is where we literally get to watch the tumor actively rewrite its own blueprint in real time, right? Just so it can evade therapy and colonize new tissue.
Exactly. Let's look at patient SJOS000111 or 1. This was an osteosarcoma case presenting with 6 distinct lung metastases.
6 different spots in the lungs.
Right, and the longitudinal sequencing revealed a clear evolutionary tree. Down at the trunk of the tree, The very beginning, they found early foundational translocations on chromosomes 4, 11, 16, and 17.
So these were the truncal SVs present across all the tumor sites.
Yes. But as the cancer disseminated, it didn't just clone itself.
It didn't just copy and paste.
No, it continued to structurally mutate. The researchers mapped shared intermediate structural variants across subsets of the metastases, and eventually they found private, late-stage SVs that were entirely unique to specific anatomical sites.
Like bespoke mutations for different environments?
Yes. In the left lung metastasis, for example, the tumor had evolved a massively complex, localized amplification on chromosome 14 that was completely absent everywhere else.
You know, we so often visualize the genesis of cancer as a big bang, like a singular catastrophic event that creates a malignant cell, and then that cell just blindly replicates.
A lot of people picture it that way.
But this spatiotemporal data proves that structural variants provide continuous evolutionary plasticity. The tumor is a highly dynamic organism. It's actively shuffling its genomic deck in response to the selective pressures of the microenvironment or even the chemotherapeutic drugs we use.
And we see an even more radical mechanical adaptation in a rhabdomyosarcoma patient they studied, patient SJRH-B0121. Early in the disease progression, the sequencing identified extrachromosomal DNA, or ecDNA.
Okay, for you listening, if you are visualizing a standard 23 pairs of chromosomes stacked up neatly, ecDNA completely breaks that architecture. These are circular, free-floating fragments of DNA that have detached entirely from the linear chromosomes.
Right, they're just floating in the nucleus. And because they exist as independent circular fragments, ecDNA lacks a centromere.
And the centromere is important because...
Well, during normal cellular mitosis, when the cell divides, the centromere is the anchor point. It ensures the chromosomes are pulled apart and divided evenly between the two new daughter cells.
Oh, so without a centromere, ecDNA just segregates randomly.
Asymmetrically, yes. One daughter cell might inherit 0 copies of that circle, while the other inherits a massive cluster of them.
Wow.
In patient SJRHB012, this asymmetric segregation allowed the tumor to accumulate 100 copies of the MDM2 oncogene on these free-floating circles.
100 copies. It's an elegant but terrifying mechanical exploit. By removing the oncogene from the main chromosome, the tumor basically bypasses the rules of even distribution. It forcefully amplifies its own growth signals to these extreme concentrations.
But the evolution didn't even stop at the cDNA. When that patient relapsed, the genomic landscape had shifted yet again.
What did it do?
The tumor had generated entirely novel structural variants fusing chromosomes 12 and 22. It didn't simply rely on the MDM2 amplification anymore. It engineered alternative survival circuitry just to overcome the therapeutic bottleneck we put in front of it.
So if the tumor is relentlessly restructuring its genome to survive, what exactly is it aiming for? I mean, it can't just be tearing cages out at random. There has to be a functional bias to where these structural variants actually land.
There is a profound functional bias, and honestly, it highlights a fundamental divergence between adult and pediatric oncology.
How so?
Well, when the researchers mapped the 10,672 genes disrupted by structural variants in this study, the adult targets were predominantly categorized as fragile sites.
Fragile sites are basically regions of the genome that are just mechanically unstable, right? Like they are prone to replication stress and physical breakage, especially as the cell undergoes decades of division.
Right. In adults, over half of the most frequently disrupted genes are simply the victims of mechanical wear and tear. It's collateral damage.
Just age taking its toll.
Exactly. But in the pediatric cohorts, the top-ranked SV targets are overwhelmingly driver genes.
Driver genes actively push the cancer forward.
Yes. These structural variants in kids are not random fractures. They are highly selected alterations that actively confer a growth or survival advantage to the malignant cell.
So pediatric structural variants are essentially precision missile strikes on the exact regulatory nodes required to drive the cancer.
That's a perfect way to put it, which leads us to perhaps the most vital clinical revelation of this entire study.
Lay it on us.
It's the discovery of a massive genomic blind spot involving this DAG2 gene. Yeah, it encodes a subunit of the cohesin complex, which physically tethers sister chromatids together during cell division.
Yeah.
Historically, we knew STAG2 operated as a driver gene in acute myeloid leukemia and Ewing sarcoma.
Right.
But our understanding was limited entirely to point mutations, those single nucleotide alterations we talked about, specifically within the coding regions of the gene.
So we assumed we understood exactly how the gene broke, and clinical assays were just designed to scan the coding exons looking for those specific known spelling errors.
Exactly. But by analyzing the whole genome data for structural variants, the researchers uncovered massive structural deletions hiding within the very first intron of STAG2.
In the intron.
Yes, specifically localized to pediatric BIL.
For you listening, introns are parts of the RNA that are transcribed but then spliced out or removed before the final protein is actually built. Because they don't directly code for the final protein, they are frequently just bypassed entirely in standard targeted sequencing panels.
They're often ignored.
Yeah.
But an intron is heavily populated with regulatory elements. And these intronic deletions were actively driving the disease. By analyzing paired samples from a patient's diagnosis and then their relapse, the researchers demonstrated that these relapse-specific structural variants within intron 1 directly cause a twofold reduction in STAG2 expression.
Unbelievable. The tumor was utilizing a structural variant in a non-coding region to actively downregulate a critical cohesion complex, which drove the relapse.
Yeah.
So what does this all mean? For a decade, we have been meticulously proofreading the text of the genome, looking for single-letter typos, entirely oblivious to the fact that the margins of the book were literally on fire.
We were looking under the wrong lamppost.
The tumor engineered its most destructive mutations in the exact genomic regions we were conditioned to ignore.
And that leads to the statistical anchor of this paper. For the vast majority of critical driver genes in pediatric cancer, the prevalence of structural variants outpaces standard point mutations by over a factor of 10.
A factor of 10.
10 times more prevalent. If a diagnostic assay is only sequencing exons to find point mutations, it is empirically blind to the primary evolutionary driver of the disease.
It's time for our final synthesis. We've covered an immense amount of ground today. We started by dismantling the myth of the quote-unquote quiet pediatric genome. We explored how germline mutations in TP53 compound structural errors over time.
And how catastrophic chromothripsis can shatter a chromosome in a single division.
Right. We uncovered the bimodal distribution of SVs in leukemia, revealing how rogue RA enzymes exploit 3D chromatin accessibility to just surgically delete tumor suppressors.
We tracked the continuous spatiotemporal evolution of metastatic clones.
And the asymmetric amplification of ecDNA. And finally, we just exposed the critical diagnostic blind spots hiding in non-coding intronic regions like in STAG2.
The immediate clinical translation of this work is really profound, because the structural variant breakpoints in pediatric ALL are highly recurrent. Specifically those 332 identified hotspots we talked about. We no longer have to sequence the entire genome in the dark.
We have a map now.
We do. Clinical genomic assays can now be structurally designed to probe these exact sub-100 base pair regions. We can identify the molecular architecture of the relapse before it clinically manifests.
It transforms the murky diagnostic landscape into a highly targeted map. It's incredible. But as we wrap up, I want to leave you, the listener, with a fundamental biological paradox to mull over on your own.
That's a big one.
We've established today that the RA enzyme, the evolutionary masterpiece responsible for generating the diversity of our immune system, is the exact mechanism actively rewriting the genome to cause pediatric leukemia.
Yeah.
It forces a really profound, almost philosophical question. If the biological tool that evolved to protect us from a chaotic, ever-changing external environment is inherently designed to introduce chaos into our own DNA, can we ever truly separate the mechanism of our survival from the mechanism of our destruction?
And therapeutically, how do you safely inhibit a process that is so inextricably linked to the core function of human immunity?
Keep questioning the blueprints of biology, because it turns out the cancer isn't just making random noise, it's actively rewriting the manual. We just had to learn how to read the torn pages. Thanks for joining us on this deep dive.